Hi everyone, welcome to our second lecture. We are making progress. Before we get started, I have several things to announce. Okay. First, I found the lecture one file was accidentally deleted by a student this morning. So I have re-uploaded the file and provided a new link in the content folder. So now the file is uneditable, so this will not happen again in the future. But for some reason, the new direct link in announcement is not working well. So it needs to be copied and pasted into your browsers. The one in the lecture links folder is fine. I apologize for this inconvenience. The second thing is that please make sure that you do not share any course materials, particularly to any public websites. These are the properties of our school, and doing so may lead to legal issue. Okay, the third thing is that for the sample test, I set it open and you can try multiple times and anytime as you wish. Make sure that you do it at least one time because our exams will be timed and any technical issue will waste your time on the exam. All right, so today we will discuss cell structure and organelle function. We will cover the page 18 to 26 in your textbook, and I will also introduce some new concepts to you. Let's first learn some history of the cells. The term cell was first used to describe the structure of a bark structure which resembles a honeycomb cells. In 1665, Hooke published a book named Micrographia, which describes the microorganisms he saw using the microscope he designed himself, shown here. One on the right side, this was the structure of cork under his microscope. The name of cell has been used ever since. After many years of continual improvements made to microscopes, scientists knew cells more and more. In biology, cell theory is the historical scientific theory, now universally accepted, that living organisms are made up of cells, that they are the basic structure organizational unit of all organisms, and that all cells come from pre-existing cells. Cells are the basic unit of structure in all organisms and also the basic unit of reproduction. Since cells are the most basic units of life, scientists try to isolate or collect cells and use them for research. They are very useful tools for scientists to understand the molecular events happening in our body. So scientists invented ways to freeze and store cells. That is using this called liquid nitrogen. Okay, liquid nitrogen is the most abundant. Nitrogen is the most abundant gas in air. So once it's pressed into the liquid form, it's super cold. Okay, the temperature of liquid nitrogen is about almost minus 200 degrees, 196. Cells can be stored in liquid nitrogen containing tank. They show like that. That cell will be frozen in there. So then inside of the liquid nitrogen tank, you can see they have different racks. Okay, so the boxes can be inserted into these racks safely. After the cells are frozen in these tubes called cryovials, they can be stored in these boxes and then stored in the liquid nitrogen tank. It has been tested that cells maintain their viability even after 30 years of storage. So theoretically, the cell's property can be kept for much longer time, hundreds or even thousands of years. Okay. What kind of cells need to be stored for future use? Actually, semen, embryos, oocytes, ovarian tissue, and then testicular tissue. These are related very useful materials in medicine. And umbilical cord blood contains a high percentage of stem cells. These also need to be stored because the stem cells, they have a differentiation potential, and they can be used to replace damaged cells if the baby develops certain diseases in the future. When moms deliver babies in hospital, usually they will be offered this choice for, you have to pay for that, there's a fee. But I think it is a smart decision to keep the cord blood. We will talk about what diseases can be treated with stem cells in the next slides. Okay, this is basically store cells needed for future use. Okay, here we listed the types of diseases that can be treated using cord blood. Currently, FDA has approved to use cord blood to treat nearly 80 diseases, including cancers and immune disorders. Please remember the best source of the cord blood is the person's cord blood. Avoid immune rejection, okay? Cryopreservative does not only apply to cells, but also for whole body or brain after people's deaths, with the speculative hope that resurrection may be possible in the future. There are companies in the US run this business as well. As of 2018, the cost of preparing and storing corpses using cryonics ranging from US dollar about 28,000 to 200,000. However, at this point, I haven't seen any report that warm-blooded animals can freeze and then come back to life after thawed. Okay. Due to the importance of cells, in 1974, a journal named Cell was published by Benjamin Lewin. Okay, so this is the Benjamin Lewin published the first journal of Cell, and this journal has been one of the leader journals in nature science. One important criteria to tell if the journal is good or not is to look at its impact factor, and it is calculated by its citations. The Journal of Cell was ranked the first overall in the category of highest impact journal over 1995 to 2005, with an average of 161.2 citations per paper. Okay, so in 2019, the impact factor of Cell ranked the first out of 298 journals in this biochemistry and molecular biology field. So this is a very good journal. There are more than 200 types of cells in our human body, with about 37 trillion cells, okay? So cells may have different shapes that can be best suited for their functions. As we learned from the cell theory, the basic structure and functional units in all organisms is the cell. So although they may look different, but structurally they are all very similar. All eukaryotic cells have a plasma membrane, cytoplasm, membranous organelles, and membrane bound nucleus. All right, we will talk about those in detail later. Cells are constructed from 4 classes of macromolecules: carbohydrates, lipids, proteins, and the nucleic acids. You should have learned all these, and I will not get into details. Okay, so this is a diagram of a generalized human cell. It shows the organization and the distribution of organelles as they would appear in the transmission electron microscope. So the type, number, and the location of organelles are related to cell function. I would like you to recognize and remember the function of these organelles, okay? So let's watch an introductory video first. It is going to talk about those organelles one by one, all right?
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants, that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Now let's discuss the structure of cells in detail. First is the cell membrane, also called a plasma membrane or cytoplasmic membrane. It is a biological membrane that separates the interior of all cells from the outside environment. It consists of lipid bilayer, including phospholipid and the cholesterol, shown here. The function of cholesterol is to maintain the integrity and the fluidity of the plasma membrane. Higher the cholesterol level, less fluid or more rigid the cell membrane would be. Plasma membrane also contains all sorts of membrane proteins that are responsible for various biological activities. Many membrane proteins are glycosylated, meaning they have a carbohydrate chain. Carbohydrates are important in the role of cell-cell recognition in eukaryotes. They are located on the surface of the cell where they recognize other cells and share information. And the viruses can also bind to cells using these receptors. Do you all still remember where the carbohydrate chain is added to the proteins? It was just showed in the video. Yes, it is in Golgi. The liquid bilayer is composed of phospholipids, which contain a hydrophilic head and hydrophobic tails. Please think about which side is facing outside and why. You can pause here if you want. Okay, so think about where cells located. Outside the cell membrane, the environment is water-based, like body fluid. And inside the cell membrane is the cytoplasm, which is also water-based. Therefore, we can imagine that hydrophilic heads should be facing outside and hydrophobic tails facing inside. This is indeed the case. Actually, the hydrophobicity plays a very important role in structural determination of almost all biomolecules. Similarly, hydrophobic amino acids, they tend to interact with each other and are essential to establish an overall structure of proteins. We will talk about that in the future. There are many kinds of proteins in cell membrane. Scientists have classified them into several major categories. I'm going to go over them quickly one by one. Okay. The first one is receptors. Some chemical signals by which cells interact, like epinephrine, it cannot get inside the target cells by passive diffusion. Instead, they can bind to surface proteins. These are called the receptors. Receptors are normally particularly for one specific messenger, similar to an enzyme that specifies for one substrate. Okay. The second one is enzyme. Enzymes in the cytoplasmic membrane can assist in production of second messengers or break down hormones and other maybe other signaling molecules once their task is done. Therefore, we're stopping them from exceedingly stimulating a cell. All right. So 3 and 4, the C and D basically. So those are the ion channels and gated ion channels. Okay. So ion channels are transmembrane protein or protein clusters with pores that permit water and liquefied ions to go through the membrane. Some channels are constantly open, like this one, and then some others are gated open. They can only open and close on the various different situations. Okay, number E, the fifth one, is the cell identity markers. So these are mainly glycoproteins. You can see this carbohydrate chain here. For instance, they can imitate a recognition tag that allow our bodies to inform which cells come from it and which are foreign intruders. There's a recognition marker. Okay. And then also the last category is the cell adhesion molecules. The cells associated with one another through those adhesion molecules. These are important for forming I think majority of the cells in our body form once they're differentiated and not invaded to other places, they're going to form these called the CAM, basically cell-cell adhesion molecules. All right, so those are the major types. Now let's move to the organelles inside the cells. I will only focus on their structures and functions and go over quickly. Okay, this slide shows a three-dimensional representation of the endoplasmic reticulum, also called ER. Rough ER has ribosomes, and smooth ER does not. Okay, so what does ribosome do? Yes, okay, making proteins. The process is called a translation, and the translation only occurs in cytoplasm. What does the gene products mean here? Yes, that means protein, or you can say amino acids or polypeptides. As long as you understand the concept, no matter how the words are changed, you can always make the right choice in exams. I would like to show you how the structure of an organelle correlates with cell function. These are the two electron micrographs of B cells. Notice that they are quite different. One is unactivated and the other is activated. Functionally, the activated B cells make antibody, which is a protein, right? But unactivated B cells does not, okay? What differences can you observe? Now, why they have these differences here? You may want to pause here to think. Okay, first, we notice that in activated B cell, which shows in the right side, contains much more ER in their cytoplasm. This is correlated with its need to make antibody by translation. Second, you may notice that the activated B cell contain large area of euchromatin. We learned from last lecture, euchromatin is enriched in genes, indicating that genes are actively transcribed in nucleus to support all the needs for the B cells to proliferate and make antibody. Now let's take a look at Golgi and lysosomes. Lysosomes are garbage cleaner and it contains abundant digestive enzymes. These enzymes are synthesized on ribosomes attached to the ER, internalized and moved to the Golgi in transport vesicles. In the Golgi, the enzymes are modified and packaged. Lysosomes pinch off the end of the Golgi membrane. In the cytoplasm, lysosomes fuse and digest the contents of vesicles that are internalized at the plasma membrane. Okay. On the right, here shows a transmission electron microscope picture of the Golgi complex. Now, here comes mitochondria. Everyone knows that the mitochondria is the powerhouse of the cell, and it provides energy to all kinds of cellular activities in the form of ATP. Remember that mitochondria only exist in cytoplasm. Okay, in last lecture, we introduced the DNA and chromatin organization in nucleus, okay? Here shows another picture of nucleus, but focus on the nuclear membrane, or sometimes called a nuclear envelope. The nuclear envelope keeps your DNA inside the nucleus to protect it from surrounding substances in the cytoplasm. The nuclear envelope consists of two double-layered membranes with the inner nuclear membrane on the cytoplasm side and the outer membrane on the outside. So these are protein complexes that spans the nuclear envelope and allow the transport of molecules across the nuclear envelope. They are named as nuclear pores. It shows like the hole here. Think about what kind of molecules that can transport it through nuclear pores. Actually, many kinds, okay? For instance, RNA and ribosomal proteins move from nucleus to the cytoplasm, and proteins such as DNA polymerases, carbohydrates, signaling molecules, and lipids, they need to be moved from cytoplasm into the nucleus. All of these are through the nuclear pore. This picture shows in the middle here is a picture of nuclear pores. On the left, the structure in the center of the nucleus is the nucleolus and responsible for ribosomal biogenesis. No matter how cloudy it is, the genetic material, the DNA, stays inside the nucleus as the form of chromatin. However, if the cells are moved to the mitotic stage, we cannot see chromatin structure anymore. So instead, we see chromosomes, as shown on the right picture. Normal human beings should have a constant numbers of chromosomes. The number and appearance of chromosomes in cell nucleus is called a karyotype. We all know that humans have 23 pairs and 46 chromosomes. Half of them come from father, half of them come from mother. Karyotype can tell you a lot of genetic information. We will have a lecture specifically studying karyotyping. But here, can you tell if this individual is a female or a male? Check here. So yes, this person is a female, as she has 2 X chromosomes and no Y chromosome. Here is another example. You can tell that this individual is a male, as he has 1 chromosome of X and 1 chromosome Y. Have you noticed anything unusual? Yes, he has 3 copies of chromosome 21. This is a typical trisomy 21, which has another name as Down syndrome. All right, so you can tell karyotype analysis can help us to diagnose certain genetic diseases. Here, I have a question for you. Where can DNA be found in a human cell? Yes, we have learned that genomic DNA is located in the nucleus, and it is composed of more than 3 billion base pairs and contains approximately 20,000 genes as the form of either chromatin or chromosomes. These numbers are important, and I hope you can remember them, all right? So interestingly, DNA also exists in mitochondria in cytoplasm. It is called mitochondrial DNA, also mtDNA. The size of mitochondrial DNA is much smaller than genomic DNA. Totally, it only contains 37 genes. The form of DNA structure is double-stranded and circular. Importantly, mitochondrial DNA normally is exclusively maternity inherited, meaning inherited from mother. Why is that? You can pause to think. This is because mitochondria are all in cytoplasm. When sperm and egg fuse and become zygote, the sperm contribute its nucleus, but barely any cytoplasm, right? And the mitochondria in sperm are usually destroyed by the egg cell after fertilization. In contrast, egg is the biggest human cell. It is about 20 times larger than sperm. It provides almost all the cytoplasmic materials, including mitochondria. Now we have learned the organization and distribution of organelles showing in this picture. During the last part of this lecture, I would like to introduce a new concept to you named phase separation. This field became quite hot in recent 5 years, but I haven't seen textbooks that have this information, all right? So that's the other function structures, including the human cell. For these materials in this part, just follow my slides, okay? Phase separation here really means liquid-liquid phase separation, LLPS. It is the phenomenon in which solutes spontaneously separate into the demixed liquid phase suspended with the bulk solvent. Have you ever tried to mix oil with water and found the oil forms the oil droplets but not dissolving water? So that's actually an example of phase separation. But why we care about it? Interestingly, as many of the organelles we learned contain cell membrane structure, there are organelles or subcellular structures that do not have the membrane structure, but can hold their specific components. We learned one of them, called a nucleolus or nucleoli. Okay. Other similar structures include the Kohala bodies, also sometimes called the CB. These are in Kohala bodies, their splicing factor can be assembled. And also histone locus body that processes histone pre-mRNAs, as well as paraspeckles that can hold mRNAs from nuclear export. Scientists have tried to generate recombinant proteins that are important for maintaining the structure of those droplets. And these proteins can form these fluid droplets in test tubes. We also call this in vitro experiments. In these proteins, if those proteins are tagged with GFP, namely green fluorescent protein, they would be seen as green under the fluorescent microscope. Okay, under the microscope in figure C showing here, you could see that these droplets have no membrane structure. The shape and size of droplets are dynamic. They have this ability to fuse together, as shown on this bottom figure. Liquid-liquid phase separation or liquid transition has been reported to link with many neuron degenerative disease. I will use the ALS as an example. ALS is also have another name as amyotrophic lateral sclerosis. It is a processive neurodegenerative disease. In the United States, it is also known as Lou Gehrig's disease, after the baseball player's name who was diagnosed with this disease. All right. So ALS affects nerve cells in the brain and the spinal cord. You probably know that motor neurons are specialized for the controlling of the important muscle activity. ALS kills motor neurons, causing muscles to weaken. Over time, ALS paralyzes the muscles used to breathe, to speak, and to eat. So on average, death occurs within 3 to 5 years after symptoms begin. But some people with ALS live 10 or more years. I'll show you an example. You probably know this person, right? So Stephen Hawking, he was a miracle. He was diagnosed with an early onset but slow processing form of ALS in 1963 and lived with ALS for 55 years. Hopefully my math is correct. In order to promote awareness of this disease and encourage donation to ALS research, the ALS Foundation introduced this called Ice Bucket Challenge in summer of 2014. And then later it became an annual event. Maybe some of you have even attended this social activity. In my last class, actually there were 2 students who participated in this activity. But what causes this disease? Scientists still do not have a clear answer, but they are making progress, right? So aberrant phase separation actually becomes one of the mechanisms causing this disease. Now let's watch a video that reports a finding that is published in the Cell Journal.
Hello, I'm Tony Hyman.
I'm Simon Alberti.
I'm Hyun K. Lee.
I'm Avinash Patel.
And we all work together in Dresden at the Max Planck Institute for Molecular Cell Biology and Genetics. And today we're going to tell you a story about how studying the dynamics of intracellular compartments led to insights about neurodegenerative disease. As so often in science, cool breakthroughs that relate to human disease come from basic research. My lab studies how the cytoplasm is organized. In our biology textbooks, we learn about organelles that are surrounded by membranes, but there are many compartments in the cell without membranes. These compartments are a way for cells to concentrate components for a wide variety of biochemical reactions. And it turns out they form through a process of liquid-liquid demixing or phase separation. We can think of this much like how oil and vinegar separate from each other in a vinaigrette. To keep this liquid state, the proteins inside compartments have to be able to slither around over each other easily. But a crucial question is, what kind of proteins have this behavior?
In my lab, we study proteins that contain large disordered regions, known as low complexity or prion-like domains. Changes in many of these proteins are associated with age-related diseases. It turns out that in healthy cells, these proteins are often found in membraneless compartments. So we decided to team up with Tony's lab to see if prion-like proteins can form liquid droplets inside cells. One such protein, called FUS, is particularly interesting because it is implicated in ALS, or Lou Gehrig's disease.
To test whether FUS forms liquid compartments, we use the following criteria. First, at steady state, without any external deforming force, liquid droplets assume spherical shape. Second, molecules within liquid droplets move around freely and rearrange. Third, liquid droplets that come close to each other fuse and become one larger droplet. To look at this, we used a new microscope built for us by Gene Myers and his lab. We found that both FOSS compartments in the cell and those formed in the test tube do meet these criteria. FOSS droplets are spherical, and molecules move around within these droplets. We also see FOSS droplets fuse, so it was clear to us that FOSS normally behaves like a liquid.
In patients with ALS, FOSS forms aggregates in motor neurons. So if FOSS behaves as a liquid in healthy cells, why does it form aggregates in disease cells? We made versions of FOSS that contain the same mutations that are found in ALS patients. However, we're a bit disappointed that in a test tube, the mutant proteins behave exactly the same as the normal proteins. One evening, while discussing this with Louise Jaworth, we realized the results made sense. Because patients with ALS live for many years without any signs of the disease. We rushed to the lab, and what we discovered was really striking. When aged in a test tube, the mutant droplets form solid aggregates, which meant mutations make FOSS undergo a liquid to solid phase transition upon aging.
Seeing this liquid to solid phase transition was one of those wow moments. We, like so many others, have asked ourselves why many neurodegenerative diseases are associated with protein aggregation. What our results suggest is that these proteins are normally associated with the formation of liquid compartments, where they can be concentrated, for example, to mediate biochemical reactions. However, the formation of liquid compartments comes at a trade-off that proteins in the compartments tend to aggregate. Therefore, in disease, they undergo a transition from the liquid state to the non-physiological aggregate. This is what we're calling an aberrant phase transition.
Many questions remain for the future. How does a cell prevent liquid to solid phase transitions? Why are young cells protected against disease? And can we identify drugs to slow down or reverse these aberrant phase transitions? It's very likely that many prion-like proteins form liquid compartments and, with age, turn into disease-causing aggregates. Thus, our findings could provide a general explanation for why protein aggregates are a hallmark of so many age-related diseases.
This slide summarizes the finding in this movie. I would like to emphasize several important points, and understanding these mechanisms will help you perform better in exams. First, the prion-like domain is a highly disordered region in FOSS protein, meaning this region is highly flexible and does not form a stable structure in the solution. This feature is extremely important for FOSS to form liquid droplets whenever necessary. Second, aging is a crucial factor to trigger ALS. The healthy cells handle aging well with forming these liquid droplets, such as stress granules, which appear when the cell is under stress. The function of stress granules are not completely known, but it has been shown that the stress granules contain high concentrational proteins as well as RNA. Therefore, they can hold the translational process of some messenger RNA. However, ALS patients may not be able to form normal liquid droplets. The mutations on FOSS is an example. So the mutated disease state and mutated FOSS form these protein aggregates instead of liquid droplets. And these aggregates will disrupt the normal function of motor neuron. Of course, there could be many reasons on how ALS is developed. This study only shows one possible mechanism. By understanding how the disease is developed will help scientists to find solutions to treat these tough diseases. This is a typical example of what the research labs are doing. I'll show you many examples in the future lectures. Lastly, I would like to introduce you an online database of genetic diseases. It's called the Online Mendelian Inheritance in Man, also called OMIM. Okay, so this database is a continuously updated catalog of human genes and genetic disorders and traits. they have a particular focus on the gene phenotype relationship. So the database document almost all the genetic disease you could think of. And it's a great source when you look for information for your team project. So this is what the website looks like. Here is the where you can search. Of course, you can search a database by disease name, gene name, or even clinical synopsis. You can play with it when you have time. Okay, so that's all for today. Stay safe, everyone, because hurricane is coming. Hopefully, I can see you guys online by Zoom meeting this afternoon. It's 4 to 5:30. So that's, if you have any questions, you can ask me at that time, okay?